Like, you're doing an astonishingly good job of operating this arm. Most people really struggle with it, but it's hilarious that the place where you melt down is which direction clockwise is. So the thing that most people have trouble with is pitch in versus pitch out.
Okay. Yeah, see, once you explain the bicep to me, that makes a lot of
Yeah,
sense now.
totally. That's
Okay.
how I have to think about it to get it right.
I've been really bad.
Okay.
Okay, I'm going to shoulder up quite a bit.
Great.
Yeah.
Can we get a little spinning?
Yeah,
Oh,
for
yeah,
Can we
yeah.
get
we
a look
get
at it
the
before we...
spinny.
Actually, that's a great place because it looks like it's in the light pretty nicely.
Yeah, do you want me to zoom in a little bit too?
Yeah, let me line
Yes,
up
please.
for you.
Okay.
Okay, go for it.
Thank you. Coming in for zoom.
Ooh, nice.
All right, want a little twirl?
It's in 2, I think.
Yeah, it is.
That looks like more crust.
It
I
may
think
just
it's
be crust,
crust.
right?
Yeah.
Well, we got
Do
it this
we-
far. Should we bring it home anyway?
Up to you.
Yeah.
Whatever you want, yeah.
Yeah,
Sure.
Is it useful?
everything's useful.
All
Okay.
Is that full?
No, I mean,
I can
zoom?
go full back.
Yeah, okay, I just need more.
All good.
Okay, and I will give you the bio box.
That would be great.
I guess we call it forward box now.
Well, bio isn't going into it, so
Yeah.
All right.
the rock box of science.
Which side do you want to put it in?
That could probably go on either side. You could put it in with the first one as well.
yeah,
Like you
I
can
mean,
tell
they're
those
sufficiently
apart.
different from
Omega.
each other.
It looks like.
Yeah, the
Can I
omega
get
side.
Yeah.
more zoom out on the...
I'm at full wide right now, so.
Oh, yeah, or just...
We're just,
You're
this
good.
is just the way.
Yeah, that's great.
If it's that hard a crust that can survive, there are wonderful things you can do. You can like drill down into them like 1/10 of a millimeter at a time, and you can reconstruct ocean history,
Yeah.
like the
Oh,
composition
cool.
of the ocean from crusts like that.
Like
Okay.
tree
like-
rings.
Yeah, exactly. Yeah, because that's exactly how these things grow. They grow from the inside
I'm
out.
going to
And
azimuth
so
right, so maybe if you can take a look at more of the bio box. I just want to see that I clear it. And shoulder up. Sorry, did we decide on a side? Do we care?
Yeah, omega.
Okay.
we have
I could
Greek bio
definitely
boxes.
Very
mess
fancy.
something up right now. Can I get a better view of the arm?
Yep.
I just can't quite remember where. Okay.
I'll keep mum unless you need a hint.
Yeah, I mean, if you have a handy suggestion, I would. I'm pitched in fully right now.
Yeah, so there's a few things you can do.
Yeah.
One is shoulder up, elbow down.
Okay. Yeah, I guess just get lined up over and just.
And you're good. There's nothing to
Okay.
hit up here.
Okay. Well, thank you. I think this will.
You don't need to come in any further, I don't
Okay.
think. You'll see as you come down.
That should be fine. Yeah, okay. I'm going to pitch out very slightly. And elbow down. Okay. Okay, I'm going to
Nice job.
put it here. There's your
Oh.
Many pieces.
There's
Cloud
rocks.
rock.
Plural. Okay.
Job.
Nice
Video,
job,
can
Ryan.
you
Thank you.
open up Iris?
job.
Yeah, can do.
There you go. I should have had done that earlier.
That's good.
Okay.
All right. Get out of here. All right. And get free. Thank you. Thank you.
Marine Science class, Orlando, Florida. This
Here's
is
the
day
view
4
up.
of 24 days of expedition. Mamanao Ngataifale, part 2. Yeah, so
And we're just
24
starting the arm
days we'll be on the ship.
Get it tucked back a little bit more.
Yeah.
Yeah, Chris, did you want to check out that rock that we saw was separated from?
Oh, yeah.
Okay,
I mean,
so just do some elbow down.
'Kay.
And you don't need to bring it all the way back. Can you pitch out too?
Yeah.
Just a bit close.
Yeah. To the
Yeah.
that.
Yeah, so some elbow down, and then it's fine to have it sort of out there, because
Yeah.
I know it takes a while to get out.
Okay. There you are. Are you good with it there? Or you
Yeah,
want-
that's totally fine.
Whoa.
When you have a good moment, could you power off Sage for the rest of the dive? Just
Power
thinking-
off, Sage?
Yeah, just thinking in case there's any salt water that does get in, maybe we
Okay.
can preserve the electronics.
Okay, sounds good.
Thanks. But no rush.
Okay, we're stable. Let's see what you can make of that.
Okay, we're going for this rock.
Is this the one we were thinking of?
I believe
Science?
it was, yeah.
That was the first one.
All right.
It might be just part of the seafloor, but Mary, I did copy that, and I'm going to turn off Sage right now.
Yeah, no rush at all, whenever you have time.
Sage is secure.
Thank you.
This is a big one.
Yeah, that's a big rock.
Big one.
You know, when you look at the fisheye camera in the top left screen,
Uh-huh.
you could almost want to believe that this just fell off the nose of
I
that
know, but
pillow.
Oh,
I think it's part of the seafloor, but we'll see. We'll see.
We'll see.
but the nose also looks pretty smooth. Like it fell off a while ago.
Well, there's a little cusp there, right? Could just come out a little notch.
There.
That's the story we're going to tell Adam.
Baby.
Only time will tell.
Yeah.
Oh, it's huge.
Oh, it is?
Yeah, you can see it with the...
It's ginormous.
It's a little sticky.
Wrist rotate is definitely sticky.
Is it? Okay.
Yeah.
Just...
No, it's a monster.
I'm actually not sure the jaws are gonna... Here, let's see.
It is loose.
Is it really?
Yeah.
Sorry, that's me just
Can you go for zoom
sticking.
a little bit more? Chris, what do you think about this rock?
It's large, right?
It is. It's not... Oh, I wonder about these front boxes. We can try. We could try.
Yeah.
But it's also going to take up our payload because it's big. And we just got to the beginning of this rocky section, so there might be other.
I think it
It's
broke.
about,
Crushing.
yeah.
Oh, is it really?
Yeah. Do you want here?
If you
I'm
can
gonna
crush it and
close-
it's just crust, then we'll just
Yeah, just crush it as hard as you
leave it.
can.
Yeah, I'm gonna close
Yeah.
a little bit. Yeah.
Oh
Yeah,
my
yeah.
Okay.
gosh.
Wow.
Right.
Okay.
Okay,
then
we don't
it's-
want that. Let's.
I can't believe there's that much crust.
That's
That's
ridiculous.
incredible. Okay, well
Okay,
that's
so
the kind
let's
of place we are.
leave that alone. And I think what I'm going to say is, you know, we have several data points now saying that even when the rocks look really good, they're really
They're
messy.
not.
You never
So I think we
know.
might just, like you say, we're only halfway between waypoint 2 and 3. My inclination is that we should stow the arm, we'll fly up and fly over these rocks, have a look at the geologic structures, and then carry on our way upslope.
Sweet.
Okay.
Great manipulator work. That thing
Yeah.
is not trivial to use.
I really like this thing. I feel like I'm at an arcade.
Yeah.
It only gets better from here,
Yeah.
honestly. Like,
My job is rock crushing. Apparently. Alright, let's get this.
Scientists do like to poke the science.
yeah,
Yeah.
that is a theme, hey?
Yeah.
What does it do when you poke it?
Yeah. We need to know.
In the lower camera right now, there's such a rad, like, pillow lava. It's just like perfectly
Oh,
circular.
yeah.
Oh yeah,
I love
the
it.
unbroken one.
I really love pillows. Actually, but my favorite feature is definitely columnar basalt.
Oh,
Like,
yeah.
pillows
Oh.
are fabulous, pizza ovens are fabulous, but like I want some columnar basalt. That's
Have
when
you
I
ever
feel.
dived on any with the ROV?
What's
Yeah.
that?
You've seen
Off
it?
of Molokai, there are some columnar
I've seen,
basalts.
like,
Really? The hexagonal
Really
ones?
incredible, like
Wow.
cathedral-like structures.
no,
They're
it's just like that.
massive. Like on cliff faces. I feel like
Mm.
off of Oregon we've seen some
Can you look over the arm a bit more?
up.
I've only seen them above sea level.
Oh, really?
There's like an island called Stafford off the coast of the Hebrides in Scotland.
Oh, yeah. There's some beautiful ones in the British Isles
Mhm.
that I really would love to see. I've never seen any on the surface. But I've seen some very
Right.
spectacular ones subsea.
Cool.
Alright, how are we with that?
As long as it doesn't show up in HD when we're centered up, it's great. I'm wildly curious as to whether we can get a Niskin.
Yeah, we can
Yeah.
try.
Yeah, when it's time for Niskins, it will be. I can't imagine we can do it, but like, I definitely want you to try.
Yeah, okay.
We'll try before the
It's like
dive
a
is
really
out.
weird
How's that?
reach.
Yeah, it's a ridiculous reach.
And when you're
One
talking about
disassembled
HD
rock.
and it showing up, are you talking about the...
We'll
which
call that
view are
one
you talking
Elon.
about?
I mean, if like the jaws were showing in the edge of
Rapidly
this,
Oh, yeah,
was
okay.
it
then
Ra-
I would
rapidly
say we still need
disassembled
to
rock?
do better. But
Yeah.
okay, let's see. Okay. I'm just seeing if it's a good idea to get another move going.
Yeah, do
We could probably
gauges.
do a 20-meter move,
Okay.
same direction,
Sounds
and that'll give us
good.
more access to this outcrop.
Sorry,
Awesome.
that was me.
Bridge, Nav.
Yeah, so Gabby, I'm going to resist the urge to ask for any more sampling, but I think if we
Okay,
just,
so-
if we just
so
hook
do
up
you
and
want
do
me
like a
to-
video
20-meter
transect
move,
over the,
150.
I just look at the, I mean, just looking at
Okay.
the geologic pictures would be interesting
Yeah,
anyway.
so you want to see more big picture stuff.
Yeah.
For your sense, like that upper cam in the upper left quadrant of my whole
Yep.
array here, that's going to be the one that's going to give you the big picture. And it's
Yeah.
shown some really awesome
Yeah.
features here. Yeah, okay. I can help
So I
you
think
enforce
just
the
a-
no sampling rule. You when there's an arm
But
in
in
front
terms
of
of
me, I'm like, no, but
Sure.
we could try.
But in terms, yeah. I mean, if we do see something that looks really, really compelling, but I think we've got our eye in now of
Okay.
Yeah, there's
Yeah.
the things that are unbreakable, and then there's the things that look like they're breakable, and that's because they're already rotting through. So
Okay, sounds good. Yeah, let's take a look. And feel free to say like, hey, can you go over this direction or whatever.
sure. But I think just like a nice slow climb and video over that. Work our way up over
And
the top until it's gone.
is there a way you need to record upper? I'm not sure how upper fits into our data stream. Generally speaking, HD is our data product.
Right.
Gosh, some of these
I'm not
pillows are
sure.
really awesome. So you might only be able to get captures with upper.
I don't know if that's that.
You know,
I think the upper camera kind of over-exaggerates it with the
Yeah.
fisheye.
Yeah.
I think
Oh, look, can we look at the fish?
I think the things you can see with the
Right
the
there
regular
above the pillow.
HD camera
Don't see it.
is the way to
Like
see.
that center pillow, there's a fish right on top of it.
Your eyes are much
Really?
better than
see
mine.
it.
Oh,
It's
I do see it. Go
bulbous,
for
yeah.
zoom video.
Going for zoom.
And then I think Chris wants a video transect, so we'll stay
Oh, wow.
sort of wide unless he asks for a particular zoom. Okay, go wide.
Thank you.
Going wide.
Little cuskeel.
Okay, Chris, we
in
are
the upper,
at your
it looks like
beck and
it's like a
call.
mount, right? So I think
Yeah.
in the upper, if you just go straight ahead, I think we're just going to basically land on top, go right
Yeah.
over the top of this, right? It's almost like there's been a tongue of this stuff coming downhill towards us.
So for this, Atalanta,
Yeah.
can you keep Herc sort of a little bit more centered up?
Yeah, definitely.
It'll help me figure out how best to navigate the mound here.
Oh, there's a lot of good rock
Maybe if we could
candidates
swing a
over
little
here.
to starboard, Gabby.
Yep.
Looks like that would be kind of straight up and over this. That seems to be the direction that the lavas were coming downhill. Yeah, that looks good. It's amazing how the pillows are all just like draped all over each other, right?
From the upper view, it looks like there's a big white coral maybe in the bottom right. It's like sticking really high up.
Oh, the whites are sponges.
Oh, sponge,
Yeah.
yeah.
And the pink ones are the corals in this case. But there have been some bamboo corals around here, and they'll be long and whip-like or sparsely branched.
This is looking good, Gabby, thank you.
Okay, great. It looks like we're sort of following up and to the right maybe a little bit. And that will
Yeah.
keep us on the edge of the mound, which is where the rock outcrop is.
Okay, and what's that? What heading are we on now if we're doing that?
It's Lake 150, so
Oh,
about
okay,
what we were
that's
doing.
pretty much the right direction to be going for the next
Yeah.
waypoint anyway.
Yep,
Cute.
and I'm looking in the sonar, and it's going to keep being rocky for a bit over
Nice.
here. And they're just awesome pillows, like broken open
Aren't
here.
they? I have to admit, I'm surprised there wasn't more stuff living here attached to the rocks, but...
The current is less here.
Oh, okay.
This is a pretty good amount of stuff, though,
Hmm.
for this depth. There could be more, but this is about average in terms of stuff.
We deserve better than average.
Well, we are kind of on the lee side, backside of this feature, so we're going to have more sediment. The current isn't as strong.
Mhm.
But we got some good things. We have farreid sponges. There's that the tall one is a Tretopleura, the long paddle-like sponge.
Here's some nice anemone here.
Yeah, we got these Pheliactis hormothetid anemones.
Can
So
we
this
zoom
is-
in on that rock that the anemone's attached to?
Yes, I can.
It's
Kind of a
tempting
weird
me.
shape.
Go for zoom.
Going for zoom.
It kind of wants us to pick it up, doesn't it?
I don't know if
It
we
Can't you
can pick
hear it
that one
calling
out.
out to you?
It does look attached there.
There's a smaller rock off to the right that you might be able to pick up. Yeah, some people call this one a Venus
think that is
flytrap
attached?
anemone, but there's another one
Go wide.
that is
Okay.
also called that.
Now, the one that
No.
the anemone is on is definitely attached pretty strongly.
Mm.
But there's some to the right. I was told by
Yeah.
a scientist I know that they didn't want to do any more
Yeah.
sampling here.
Yeah, I know. No, it was just.
But I don't want to talk you out of your samples
It was just
too
that.
much.
It was just that one. Just looked like it was the kind you
Yeah.
could get the claw on.
Yeah,
shake it, but-
we definitely can try. I would not want to discourage you. Can you come up, please?
Yeah, that's what
I just don't
Yeah.
see you breaking off a piece of that rock. The jaws are strong, but I don't think we're that strong. There
It's
were
a
some other
quest.
really good candidates back the way we came.
Yeah, not going back. Never go back. That's not exploration if you go back.
Chris, do you want to give this stuff a try here?
I would love to see if we could
Okay,
get some of that. Yeah.
let's do it.
With the apologies to the anemone that's going to come with us.
That's really ambitious.
You're going to take its home.
This
And
round one looks like a better size, though.
You don't think that's just
Yeah,
crust?
there is also this one.
Yeah.
That
I was just wondering if that's just
Yeah,
a ball of
that's the
crust.
round one.
maybe.
Do you want to do the round one?
Sure.
Okay.
Try the round one if
Okay.
it squishes. But yeah, if we set up where we can reach both. Actually, this one is like one of those lava buds I was talking about before. So either one of those, if they have rocky integrity to them.
Okay, you know the drill.
All right.
This is definitely Orion's sampling day.
Yeah,
Lucky arm.
it's biceps day.
Yeah, exactly.
Yeah, well, that's a cool looking.
The anemone is really neat.
It's distracting. Yeah,
Eyes on the prize.
yeah,
Getting
I
around.
saw what you're picking up.
I love how the arm is blocking the enemy perfectly.
Down in front.
Every time. All right. We're waiting for this round guy in the middle, right?
Yeah.
Yeah. This guy? Yeah.
Yeah.
And if we can't get that, snap that bit off. But
Gabby, you're watching Atalanta, right? A little bit? Yeah. Okay. Yeah, definitely. Alright, let's see. I'm actually gonna do it more like this.
Megan, while this is happening, can you tell me the range to waypoint 3 from here, please?
Yeah, sure. Looks like 150 meters.
Okay.
Sorry, I know I'm moving us a little bit. Just trying to get in there.
Oh, it's
Yay.
loose.
Nice, okay. Just gonna try to flick it out of there. I don't know if my jaws are actually big enough. Here, let's try. Yeah.
Oh, that's
Yeah, I was like,
yeah.
"Ooh." That was
You know, when I was saying like
definitely
throw it up
totally
and juggle it
intentional.
before, I was like, I thought I was joking. But
Okay, well, it's getting less cool. Yeah, okay.
beautiful.
I think we're just I'm a little worried we're like caught in the jaws on the one side. No, okay. I'm gonna try, let's try this.
And Gabby, do you have any qualms about sample size for this, for payload?
Yeah.
Okay, good.
Jaw close. Maybe?
How hard can you squish it?
That was pretty hard.
Well, then it's solid.
So I think I
Nice.
actually have it right now. Maybe? Come on.
You've got
There
it.
we go.
Sorry, I was muted there, Chris. This is a fine size.
Okay.
It'll fit in the box. I'll get a chance to check. We have another plate to pitch too.
Okay.
And I'll get a chance to check our buoyancy after this, so.
Okay, thank you.
Just gonna close. Yeah, okay, those jaws are very closed. Should we feed it to the Venus flytrap over there?
Oh, it will fit right in
Just
there.
let it swallow it. Yeah.
All right, we're
Like
trying to
little
get it in the
Sapa
bio
horse.
box now.
Yeah, rock box.
Rock box. I am going to. Can you just show me the I just want to see the bicep. I don't know where I am right now.
Chris, is it going to make your life easier if we put it in the other side?
Yes. Yeah, if that could go in the lambda side, that'd be great.
Okay, I'm
Also, make
coming
sure we don't
over
end up
on
with a list like
azimuth.
Nautilus had
Yeah.
in port.
Yeah.
So there's
Right.
another thing you can try
Yeah.
that you haven't tried yet, which
Yes.
is to come into the box like this. It gives you
Okay.
a little bit, I don't know if it's going to work with this, so do a forearm roll
Yeah.
other way, so that you can see,
yeah.
so you can see the bicep. And then curl the bicep in, like this,
Yeah.
stop there,
Okay,
curl it in, pitch in,
We have a question on chat.
and then you're going to end up with
Okay.
a
What
are
little
we
bit
studying
more
on the
of,
rock
oh, that's a
samples?
beautiful view, of an shoulder
I think
down,
it's
elbow
mostly
up
going to be
situation.
on the
Yeah.
lava
This- and
composition.
then
that
This is going to
from
just give you some more, a
uh,
different set of
Adam's the guy
options.
who's
You
the
can
real
kind of
guru
get
on
yourself
this
sort
stuff,
of
but
stuck when
if you
you're
look
up
at the very
over
Yeah.
the
detailed
top.
composition
But when you
of
come
the
in from the
lavas,
side,
The
it tells you
history,
something about
how
where
you get a
it
little,
melts.
you get more options.
So I can drop it
Yeah,
more
but it
like
also
You'll
tells
drop
you
it
like how
sideways,
deep
yeah.
they came from within the earth, because the lavas
We'll give this a
here
try. I
come
don't know
from
how it
much
works
deeper
with this
than
arm,
most
but
of the
it's something you
volcanoes
do.
that make up
It's
the
the only
seafloor.
way to do it with the other arm.
Okay.
And they're long-lived, and they come from the lower part of Earth's
Right.
mantle.
Do you want to do the wrist roll
So they
here
kind of
and
like
show
have a lot of secrets to them, and they
data?
maybe
We'll
evolve
do
through
the
time
captures now.
as well. So
Yes, please.
Okay,
Sounds
go
good.
for zoom.
Going for
each
zoom.
of the islands are a different
nice rock.
age, and so
It's
it's
beautiful
looking at
rock.
how the composition has
that's
changed
good.
through time.
Go for wrist
Might
roll.
tell us about how deep the lava's been coming from.
Got some friends on that rock.
Oh, that's a pretty rock right there.
Yeah.
Okay,
All right.
go half zoom.
Half zoom.
That other one we left behind now. It's sitting there full of
Okay,
regret. Good.
let's
It could have been me. It could have been me. I could have been famous.
That's a
The
great zoom
anemone
level
has
right
no
now. Thank
regrets.
you. Okay, I'm gonna go box out.
It's like
Okay.
Richard Dreyfuss
Yeah,
in
fine.
like Close Encounters, right? It's like, I want to go for the ride in the spaceship.
Can you elbow up just to make sure that I'm gonna clear you?
Yes.
There you go.
Nice.
That's good.
Alright.
Okay. So this is sort of exploratory, but I think you're going to find that with your pitch in,
Yeah.
you're going to be able to do this with azimuth,
Yeah. Okay.
and it should give you a lot of options.
I see what you're saying here. I just see if I clear that.
And
Hey.
so we may have to rotate the rock.
Yeah.
So the long axis the other way,
Yeah.
so that the long axis is
Like
yeah,
that.
there we go. A little more.
Yeah, it's just sticky. Sorry.
Yeah, totally. I got that.
There we go. Yeah. Okay.
And
And-
so you're going to lambda there.
Oh, okay.
Yeah. This is a bigger one. Oh, actually, science, before we put this in,
Mm-hmm.
so this is probably going to take up most of lambda,
Yep.
which is going to leave you with just omega for sampling for the rest of the dive,
Okay.
because we can't access the starboard bio box right now.
Oh, okay. Got it. Yep, that's fine.
Okay.
Can you still suction sample?
Interesting question.
Oh, yeah, that is a good question. It's like the Niskin question, right?
Okay. I love this. So if
Actually,
I...
and Megan, if you think this is not specially sparse in terms of biology, this might be a place to get an eDNA Niskin sample,
Oh, okay.
since we've been loitering in. How many Niskins are there?
6.
Oh, yeah.
Yeah.
I think there's
We
a
can
decent
totally
amount of
afford to
stuff
find a
around.
1 then. That's not extravagant.
Yeah.
That'll be all right.
No.
Yeah.
If we can do it.
You know,
Yeah,
while
that
we-
is unknown.
Yeah.
So one thing you might try
Yeah.
is getting the edge of that rock sort of on the edge of the acrylic,
Yeah.
and so that it doesn't roll in, but instead slides in.
Yeah, okay.
Does that make sense?
Yeah, I'm just trying to figure out which ones get me kind of
Yeah.
there. And it's,
And we might need to like tap it around a little bit, or
yeah, okay. Sorry, it's
You're doing a
it's
great
actually
job.
more azimuth and stuff than shoulder.
yeah. And
ended up
actually, we
being,
can
but
tone down the azimuth if it's too spicy.
It's fine. I just don't click it as much. But yeah, good to know. Okay.
Yeah, all that stuff's adjustable.
I think.
So don't come down any further.
Yeah, I'm gonna.
And when you open, you actually probably need to come up a bit, because when you open, it's going to...
Not let me
did you
out.
see how you just
Yeah.
tapped the acrylic there?
Yeah.
But you shouldn't need to open much to get to let go.
Yeah. Okay. What do we think? Open?
Sure.
Here, I think that's about.
Beautiful
Oh,
job.
that was
Nice.
amazing.
Yay.
That was very satisfying.
That was an
was
awesome
really
sample.
satisfying.
Yeah.
3 points.
That's
Yeah.
sample 5.
Yes, 005.
All right, I'm free of you if you want to
Okay.
go in with the bio box. Okay, jaws closing.
So, did I hear that you want to attempt a Niskin?
Yeah. A couple of yoga stretches and then a Niskin
Yeah,
reach around
let's limber
the
up
right
here.
shoulder.
Ugh.
Where are they? Where?
Okay,
The
so
little
see
golf
those
balls.
lottery balls there?
Yeah.
Those are how we, so the Niskins are on
Oh.
the port side, and the lottery
Ooh.
balls are how we trigger them, and
Yep.
they are exactly in the armpit. So,
Yeah.
I have no concept of whether we can do this, but we will sure try. So your best view of the relationship between all these items is upper. But so you see in upper how like
Yeah.
it's like it seems like it's kind of close. And yeah, I don't know. It's I'm not optimistic, but I
No.
still want to try it because it's possible.
Yeah.
It's a challenge, right?
Yeah. And the best way to get a pilot to do something is to tell them that you don't think they can do it.
Yeah.
It's magic. Yeah, I don't think you can get that sample. It looks
It's like,
too
that's
hard
not
to
gonna happen.
reach.
Yeah.
Yeah, exactly. Those are the magic words.
So,
Yeah, what do you think?
yeah, I'm just trying to decide how. I'm wondering if I came in. I'm just trying to remember how far this goes. Like that almost. With.
So you have a view in bubble.
Yeah.
You have a view in upper.
Yeah, and I can see over there. Okay, so remind me, actually, yeah. Sorry, my brain just realized I've only ever hooked up the Niskins on deck, and that's not where we're going to trigger.
That is not where we're gonna trigger them.
Yeah, I was like, all right, so we're going for all these colorful balls right in
Yeah.
my face, right? Yeah, okay.
Yeah, and so the only hazard is you are going to be coming in on azimuth, which is
Yeah.
kind of hot, and you're going to be coming in towards the HD.
Yeah.
So first thing to do is to shoulder all the way up.
Okay.
We can also we'll turn down azimuth a bit, because that'll give you a little more control. I'm just
Okay,
turning
yeah,
down
that would
azimuth
be-
right to
That'd be
10%.
great. Okay, we're all the way up, shoulder.
Okay, and so now I think azimuth, I would azimuth right and start to see how,
Yeah.
Can you explain? One of the viewers is asking, what is azimuth?
so azimuth is when we move the arm right to left. Azimuth is also what you call the positions on the compass. So it's basically, another term for it would be, could be yaw.
Interesting.
But
Oh,
we call
you turned
other
it
joints.
down. That would make
Yes,
sense.
I did turn it down. But we call other joints yaw, so we call this one azimuth.
Thank you.
Are you shouldered all the way up?
Sorry, I went out because I thought I was stuck on my own. There we go. Just a little.
Yeah, you should be fine.
Yeah, I see it, and then we'll pitch
Okay.
in and try to.
Yeah, and actually, so it might be like a pitch in and a forearm rotate
Yeah.
down. It's gonna be interesting.
So which one are we just trying to trigger? Any of them or all?
Yeah.
If we can, so now, before,
One, and they can be fired
so,
anytime, any
yeah,
order, right?
I think that we're most likely to be able to get the lowest one
'Kay.
with the strategy that I'm thinking about,
'Kay.
which is with elbow down
Yeah.
a bit, forearm rotate down, and then just have this sort of like off angle, like come in like this.
Yeah.
You're like scratching your rib cage here.
Yeah, yeah, yeah. Okay. So I just don't want to. So that's right into the.
Yeah, but you're still a little ways off.
How can you tell?
So I'm looking in upper there,
Yeah.
and you're still outside the bio box.
Okay. And this is now.
But you
tucked
can
into
do
the-
your forearm rotate down more until,
Yeah, I
yeah,
want to be
do you
like a
see?
little bit.
yeah, you want to, because when
Yeah.
you actually pitch, when
Yeah.
you go to elbow down, you
Yeah,
want to,
I will swing
yeah,
right into
now
them.
you'll swing into them. Yeah.
Do you want video to pan down just so you can see the
Yeah.
lower balls?
Yeah, that'd be great.
There we go.
We'll see if we hit our own internals here. I'm gonna pitch in, just get a
Yeah.
steeper angle here.
This is so ninja.
Yeah, and you will run out of the real our limiting factor here is you're going to run out of pitch, which is why I don't know if we'll ever get much higher than 6 or 5.
Yeah, okay.
But,
yeah, because that's all of our pitch.
okay.
And I'm just gonna...
And so now it's elbow.
Yeah. Okay.
And it's also going to be hard to grab these.
Do you,
I really cannot tell if I'm just gonna hit my own... okay,
so
I'm on the outside of it?
yeah, you are. And then you're going to need a little counterclockwise probably. And I'm only going by upper. I only really have upper right now. But
yeah, I can see because I'm not quite
so
lined up.
another place you can see it is probably bubble.
Yeah, okay.
do you need to rotate the claw before you go in there as well?
Claw might be
Unclear.
okay, actually.
Okay. I was just thinking you might foul against the upper part of the arm, like up
Could
the
be.
shoulder, whereas if it
Could
was
be.
rotated, it
Yeah.
could just like
I
open and
like
close in
that.
alongside.
Sometimes we actually trigger these without opening the jaws
Oh.
too.
Okay.
Yeah, I see what
Where you catch them from the bottom or the side and you just pull up.
Oh, okay.
So that's one way to do it. So this is number 6. So I'm gonna do. You can keep sort of honing in on what you're trying to get to, but I'm gonna get some different views happening here.
Yeah. Nice. Okay, that actually lets me know where I am.
That's not what I wanted.
Nice.
So
Wow.
now you've got bubble down here.
Oop.
Oh yeah, great.
Sorry,
That's looking
going
good.
a little too
Okay,
far.
so before you pull it,
Yeah.
we have, I want to get a view, the port view there that we have. I want that in a place where people can see it, like maybe Mitch hold here. Okay, so now we can see when a Niskin triggers.
I think I gotta go a little bit more. Okay.
Okay,
So we are good to try to pull on
science,
it?
are we ready for this?
Oh yeah.
I think
Okay,
so.
let's do it.
Okay, we might not get it on the first go here. Let's see. Oh, I think I actually do. No.
Yeah, give it I think you had it.
It's there we go. Okay. Nope.
And Data, you're watching, this screen here to see when it triggers?
Yep.
Okay.
Oh, nice. I did have a little more pitch.
And
Sweet.
folks at home can watch
Lovely.
satellite feed 3 if they want to see a Niskin bottle.
Oh, that looks perfect.
Beautiful.
Okay,
Nice.
let's do it. Okay.
I need a picture of this for Dan because he'll be so
Oh,
stoked.
he's gonna be so
He's
excited.
gonna lose his mind.
I should send it to Danny too.
Oh yeah. Okay, go for it. That's it. Okay, stop.
Okay. Sorry.
Yep, let go. Just release the jaws because if you pull too far, it
It'll snap.
breaks it. Yeah.
Okay.
While we're here, let's do.
Awesome.
Can you pitch in
Nice.
at all? Let's see. Let's explore the limits of this.
I got no more pitch.
Okay.
What I could do is I could wonder if you could hook it with.
Oh yeah, I think you might be able to. That would be a little bit. How open do the jaws go? Yeah, so
There.
it looks like you might be able to get 5, but probably not 4 from here.
Yeah.
Cool. Nice.
It,
Okay.
That was awesome.
That is a first. We have never triggered a Niskin with the Orion before. That was super rad.
Yeah.
Okay.
Good job. Thank you.
Yeah, that was great.
And Nav, that was number 006.
I'm going to go back to our original
Yeah.
camera setup.
Okay. I'm just gonna try to get out of the area of that camera.
And
It's
Chris on data
possible
just
that Dan has triggered a Niskin
to
with
let
the
you know.
Orion when
Steve
nobody was
Auskovich
looking.
wrote in on the
It
chat
sounds
just
like
to point
something
out
he
that
would
he
try to
looked like
do.
there were some attached animals to that rock
I think he
sample as
would tell
well.
us
So
about it though.
be
Yeah,
worth
you
picking
totally
over
tell
that.
you.
And he also saw some, might be some brittle stars that might have come off, so it might be worth looking through in the bottom of the box as well,
Okay,
just to
All right.
see if there was anything attached
got it.
that
Thanks.
becomes detached on the way up.
I'll keep an eye
So
out
when your
pull 'em
team are
out.
doing basket scavenging later,
See all kinds of fun stuff.
it's not just a rock.
Yeah.
It's just a biology delivery mechanism.
Yeah.
Over here, shoulder up.
Well, I'm glad we stopped. Thanks for overriding the watch lead, Gabby. He said one thing, but we know what he really means.
All right, is that going to be in your view too much?
I don't know.
Pitch down
You can pitch out,
or out?
but yeah.
There you go. No more.
The thing is, there's like this balance between like getting too many samples and getting your samples before there's no more rocks, or we have to call the dive, or, you know,
Yeah, once
the
we
weather
get
comes
shallower,
up, like, yeah, who, you know.
there won't
Okay.
be any more rocks after we get too shallow.
Is that true?
Yeah.
Oh, just because of the sediment, or?
The reef.
The reef, okay. Great, so we've got a rock.
We've got an
We
eDNA bottle of water.
We got a bottle of water.
We may have some life attached to the rock.
Cleaned up all our toys.
And we did Orion yoga.
Yeah, that was pretty rad. Okay, so I'm going to do a quick buoyancy check before we head out. Make sure we still float. Oh yeah, we're great.
Should we keep moving?
Give me 1 sec. I'm still fussing with the Z-bias here. Whee! That's the bottom. Might be a bit heavy.
Okay.
Morning.
Okay. Yeah. Onward?
Onward.
Do we want to, Chris, do we want to ship move? Do we want to keep taking it slow around here?
No, I think it's time to head to waypoint 3.
Okay,
Okay, let's make it happen.
let's go 160.
160, roger.
Bridge nav. Let's make a ship move, 20 meters, 160. Thank you. Did you see that little crab in the crevice?
Mm-hmm.
That was one of those ones that carries the anemone.
Oh, cute little backpacker crab.
Mm-hmm.
Yeah, so the logic, Gabby, is that once we got one good rock, chances are all these lavas came out at the same time. So
Oh,
in terms
gotcha.
of when we get the composition, we don't need more than one of those. But then with Megan's point that it's not that like bad for biology here, then the eDNA would give us, since we invested the time, it was a good place to get co-located eDNA data.
Okay.
And so that's what the Niskin is
So-
going to give us, is the ability
Okay.
to find out.
There's pretty good diversity here.
Yeah, so we got the species that Megan can recognize already, right? And then we can see, okay, how many of those show up on the eDNA analyses when we get back to shore? And then if there's anything else that might be loitering in the water, right? The things that might have left their aura behind. But you can't actually see them in the rocks, but doesn't mean they're not here.
So you think that this is all pretty much the same event,
Yeah,
and anything you do at the tail end, so we're at like sort of the distal end of the flow,
yep,
is going to be pretty representative
of the whole flow,
the
right?
flow.
Looks like we have a another kind of sponge.
What's this coming up in an upper?
right
Yeah,
on the
that looks
bottom there.
spectacular.
Ooh, we should look at some of these corals.
Okay.
The pink one. I'm pretty sure that's going to be a hemicorallium.
Go for zoom video.
Going for zoom.
The nice thing about being a little heavy is everything's a lot smoother.
Mm-hmm. So the pink corals are considered precious corals. They have a calcium
Stop there.
carbonate skeleton that has that pink color. Some of them are more of a white than a pink, but they can range in color from being like a really dark red
You can push
to
in further?
a light pink or
Pushing
a white.
in.
The
If
way to
this-
tell the difference between pink corals and bubblegum corals is the polyps on the ends of the branches. If they're paired, it's a hemicorallium. If there's more than one or sort of a ball of polyps on the ends, then it's a paragorgia or bubblegum coral.
If this is a particularly like unusual area with corals, then at some point we should take, when we come across particularly unusual areas, we can take discrete eDNA samples that aren't part of the standard transect. But I don't have a feel for if this is like the most interesting corals we're going to see or not. I don't know if you have an idea about that.
Go wide.
Going
I
wide.
mean, my feeling is it's going to get more interesting
Okay.
as we get a little bit shallower. Oh, there's
There's
a
a
real
cool thing,
Venus
right?
That's very
flytrap. There you
cool.
go.
Wow.
Okay,
Those
well
are
let's
so
hold off
cool.
on the like special sample
Yeah.
and wait
Yeah.
till we see something
Go
more
for zoom video.
Zoom.
abundant.
Stop there.
Stop.
That's quite nice.
That's really cool.
I mean, the good thing is we did just get a Niskin that's close
Yeah.
enough to
This-
here for
This
the
anemone
low
is called
resolution.
Actinus
It's not as
scyphia.
The Niskins only really work from
So
what
this is
Annette was
like
saying when you have
what is
really
really high
abundant.
called the
abundance.
Venus
And I'm not
flytrap
sure how
anemone.
high abundant this is in terms of
Right.
whether we'll get a signal. But we're sort of coming up to be due another of our
Okay,
depth
go wide.
points in the next 20-ish meters. So we'll probably still have the same type of ecosystem when we start the next depth gradient. Oh,
Ooh,
cool.
There's the sea
There's a sea
cucumber.
cucumber.
Haha.
It probably felt us coming and it did some crunches, but then it got tired.
I always think of them as the bison
Can
of the
you
deep.
show me the vehicle on there? Cool, thanks. And can you give me a DVL reset?
Yep. Sending. Bridge nav.
Yeah, if we're lucky, this
Can we do
ridge will
another
just
20
remain
meters,
exposed rock all the way up.
160?
So
I was going to check
Thank
what was the
you.
last depth I took an EDNA.
I'm I'm sorry, Chris, did you just say something?
Oh, sorry, I was just talking to Mary about
Oh, okay.
DNA sampling strategies.
Okay, great.
I was saying if we're lucky, then this actual feature that we're driving up on the topography will actually remain pretty rocky all the way up now. It seems like every time we go past a sonar target, then more appear on this orientation. So
We are hitting that like steeper part of this ridge, so hopefully it'll stay hard and interesting the rest of the way.
Yep.
Go for zoom.
Going for zoom.
This is a much prettier example of the hemicorallium.
You
And
can
then
push
we
in further.
have a
We should see the polyps.
xenometrid crinoid on there,
Go
and
for
then
it.
those
Yeah, nice.
yellowy polyps are actually a zoanthid.
Getting
So
the
a
focus on those
parazoanthid,
polyps is
yeah.
really tricky because it's you sort of have to pick a plane to focus on, and then I move.
It looks so edible.
Is the, Megan, is the white color something different?
Yeah,
Or is
Okay,
it
go
just
wide.
like a
Going
decayed
wide.
part of the
Nice job.
coral?
That was beautiful.
those white polyps are a parasitic zoanthid that is
Okay.
living on the coral. And so my thought is what happens is part of the coral dies and then it gets populated by those zoanthid. Oh, and this is an example of the bubblegum coral right here next to it. So now you can see what the difference looks like.
Go for zoom.
Going for zoom.
And this guy has a snake star in the branches.
So tell us
You
what we're
can
looking at
push
with the
in
polyps
further
here
and
to
get
tell the difference.
polyp zoom there.
Okay,
Sounds
see how
good.
the ends of the branches have this sort of round ball of polyps, so more than 2 polyps at the end. And tends to be a deeper red color. And the other giveaway is at the base, you see
Okay, go
how
wide.
it's a little bit
Going wide.
yellow.
Oh, yeah.
Because this animal does not have a calcium carbonate skeleton. It's more of like a protein complex.
Oh, okay.
Yeah.
And the other one is a carbonate
Yeah, it's carbonate.
stem.
I really like the snake stars. The snake stars are almost always associated with the paragorgia, but you can sometimes see them on the hemicorallium. But the hemicoralliums are very fragile and they'll break very easily. The paragorgias are more bendy. They'll move in the breeze.
Little chrysogorgia maybe?
I think there's a...
Oh yeah, there's a little chrysogorgia.
Go for zoom video.
Going for zoom.
Yeah, we have some nice
This might
life
be
down here.
There's some good stuff. This might be Chrysogorgia stellata. And oftentimes they have little squat lobsters that live in the branches.
Go a little wider.
A little wider.
There we go. That's better. Yeah.
I don't see a squat lobster in this one. That's a great view. That'll be good for identification. It looks like it has double planes
Oh, yeah.
instead of just like one
Okay, go
branch
wide.
that branches out. So it might be Chrysogorgia chrysaries and not stellata.
What's the advantage of having like those 2 planes of materials? Is it just like a more effective filter feeder that way or something?
Well, the Chrysogorgias
Do you
have
want
like a lot of different.
keep the ship moving, or do you want to wait until Atalanta settles out?
We do have a lot to see here, but we can keep moving.
I mean, we'll still have time here, even after you put a ship
nav.
moving. Oh, sorry, we're gonna do a turnover, so maybe not. That'll just hold off.
Never mind. Didn't realize we were almost ready for turnover.
Yeah.
We're gonna hold here.
Okay.
Thought it was a little early.
Yeah.
15 minutes
yeah.
early.
was fun. Yeah, I think Pilot has access to panning up, so we might have to ping Pilot on that. One sec. It's just yeah. We have some interesting bio according to Chris. Wants a pan up on the
Oh,
Hercules.
Actually,
yeah,
while
of
you're doing the
course.
turnover, we could just
Yeah,
maybe.
I'll definitely pan up. Is that what you want to see?
Cool. Yeah.
Cool.
Thank you.
Yeah, I'm trying to think about any.
So we've got a lot of sponges, different kinds of sponges in this area. One of my favorite ones is the long-stalked one with a little poof on the end.
Uh-huh.
It's called
Oh, yeah.
Caliphacus.
Looks almost like a dandelion, right?
Yeah, kind of.
Like the dandelion when it's gone to seed. Is that the one you were talking about? Like
Yeah,
with the
yeah,
little like white spear at the top?
that's the one. Except if you blow on it, it won't
No.
do anything. But sometimes they come in multiple poofs where there's like lots of different little mushroom heads, because up close they
Oh,
look
on a
like
single
little white
stalk?
mushrooms.
Oh, wow. I've seen individuals like that, like the one we're looking at in the distance. But you're saying like a single stalk can have like multiple of those little spheres at the top? Well, this is actually going to be a great timing for Annette and
For the next
to
eDNA
come look
sample.
at.
Yeah, because I think
Yeah,
it was
because we can
about,
say, hey, we've got a Niskin, but we're in amongst a bunch of stuff, so.
well, and also from my little like doodle, I was thinking that about 1320 at the central point of the next
What is
depth
about?
gradient. And we're at 1350, so I was thinking start at about
It's probably
1340-ish and
right.
go through that point. But they can then make
Yeah,
that
yeah.
call. That would be good.
Yeah, I mean, if it might well be.
It's
I
right
mean,
there.
I mean, this is there's more biological activity here than we've seen previously, so
Yeah.
it
Yeah,
would be
definitely.
different, even though it's
Yeah.
even if the depth isn't that different.
To be honest,
We've definitely gone across a gradient, I think, of
yeah.
just because there's rocks, I think, and it's better habitat.
Yeah. I mean, actually, this handover would have been the perfect time to do a discrete sample because it would have been 20 minutes of sitting still with nothing else going on. But too late. And maybe not worth it for only 3 of them.
I mean, it feels like it's changed in biological density since we got that sample.
That's true.
So even
but,
where we were sitting, it wasn't as abundant as here. So
that's true, but then Megan was saying she expects it to get better throughout the rest of the dive. So
well, I guess that's why it's perfect that it's Annette and
yeah, they can make
Santiago
that
now, because they can
1
sit
to
there
4
and say,
judgment
okay,
call.
how many of those sit still and
Yep.
samples do they want to do?
Yep. Yep.
And they do it for a living, right? This is
Yeah.
my first time of doing that kind of stuff, so they can sit
Yeah.
there and go like, oh, this is wonderful. Did you get a sample yet? Or
Yeah.
they might just sit there and go like, why are we sitting here?
Yeah. Yeah.
And we will learn from their reaction.
Yeah.
It's always the first thing, like the first watch of doing other people's science of these things is you get calibrated of
What they want.
what
Yeah.
what's exciting for other
See you in a
people. I think it was like 9 p.m. local. Yeah, me too. What's really funny is that Sean spent the whole of the first part of the cruise telling me what a light sleeper he was. He was sleeping through the same 2 alarms I was. Yeah. Trying to work out what my next system's gonna be. I'll have to have like something that's gonna pour a bucket of cold water over me or something at like 11:15. Oh, you know, do you remember when we were flying in and there was like a do not disturb mode? I need to go check my phone and make sure there isn't something going on in the background that's... Well, I even have like my wristband. I mean, I never normally sleep. That's vibrating. I mean, it's like... I guess I'm just still getting on the right sleep cycle. I should definitely go do the power nap after watch rather than just stay up. Oh, actually, no, I better not. Victoria's expecting me on a Zoom call.
Were we setting up for another rock sample?
No,
No,
we're done with rocks.
We're, I think when Annette and Santiago come in, we're kind of like, we're kind of on the cusp of whether it's time to start doing some new eDNA sampling.
Yep.
And the value judgment will be, do they want to do that as a survey or do they want to stop and sit somewhere?
Yep.
But I think we'll hold off on that till they get here. But in the meantime, we're just, I don't know, Megan, do you want to hold off till the complete watch change before we put in the ship move?
Yeah, we're gonna
Just rotate
turn
through
over
everybody.
and
Okay.
yeah, let the new navigator get settled before we move.
Okay.
Yep.
The other thing that will please Santiago and Annette is we can tell them we've done all the rock sampling we need to do.
Yeah, I think you
'Cause
have
they
space
will-
for one more in a box,
Yeah,
maybe.
but
But no more sediment clouds. No
No
more.
more.
Yeah.
It's not a priority. If they don't get another rock, then
No.
Adam
We've
will
got
be
plenty
Adam
of
will be
rocks.
on after breakfast. So if there is need for another rock, but in the meantime they can just enjoy themselves of
Yeah.
do the stuff they're excited about.
Yeah. They've just started discretionary Niskin, the first discretionary Niskin, which is number 13. Sorry.
All right, good morning, everybody. We just had our shift change, and this is now the 4 to 8 watch in the van. Give everybody just a second to get settled, and then we'll start to introduce this watch.
Okay.
Okay, good morning, everyone. This is Renato Kane. I'm the navigator here, filling in for Gabby as we train. Just to let everyone in the back row know, situationally, we are between waypoint 2 and waypoint 3, headed upslope to our final destination. So as of right now, the ship is holding station. We're stable here and ready to proceed forward or hold here, depending on your desires.
Copy that. Thank you for the update. I think we want to keep making ways towards waypoint 3. Since we're starting seeing this terrain, we want to see a bit more of what things look like here, and then we'll probably start thinking about sampling in a little while.
Roger that. Tito, how's everything going over there? You're stable?
Yeah, good morning.
Good morning.
Ready to get underway?
Great, yeah. So as you kind of strafe over there to your starboard side and line up with Atalanta, I think we're going to be moving, let me get your bearing here when I find my mouse. So we'll be going upslope, about 150 will be our, 150 will be our heading. So if we get, as soon as Hercules comes around, we'll get Atalanta's heading to 150. I'll start the ship move now. We'll start slow. Maybe we'll go with 0.2 and then bump up to 0.3 if we're not stopping and sampling too much. Bridge, nav. Good morning, Martina. Can we step 50 meters, bearing 150 and speed 0.2? Thank you. So our depth right now is 1,300 meters, 1,350. So this move should not take too long for Atalanta to see its effects. What's up? Yeah, I think you were using it for this, yeah? Yeah, you're all good. And we can, if you want, we can toggle back once we get comfortable here, back to the original and kind of compare and contrast the regular dive. It does rearrange all the small ones, but you can see it kind of allows for that 360 upper cam for more situational awareness than the lower, or than the Herc main HD. And then cam upper is just unfocused right now, but it's not as helpful navigationally anyway. Oh, sorry.
Rennie, did did you want to line up
There you go.
Atalanta and
Yep.
Herc?
Oh, 1 second. Sorry, Tito?
Do you like the two vehicles on the same heading?
Yeah, as we go up, and then you can just kind of wag back and forth, inspect whatever the scientists want, and we'll just, point 2 is such a slow crawl, we'll just kind of see, I think we'll have time to stop and smell the roses kind of thing.
Roger that. Can you hear me?
A little closer to your mouth.
Hear than that?
Yeah, better there, yeah.
Thank you.
Maybe even a little closer, actually. It's kind of like got to be right up in your mouth. Thanks. I'll crank up. There we go.
Okay, now we're ready.
Santiago, can you kind of introduce yourself and kind of what our objectives are for this portion of the dive?
Absolutely. So good morning, good evening, whatever you are.
Whatever it is.
I am Santiago Herrera. I am a professor at Lehigh University, and I'm a benthic biologist, so I study animals in the deep sea and their ecology and evolution. And so I'm the watch lead for the 428, and right now we're diving on a ridge feature north of the island of Tutuila, which is the main island of American Samoa. We started at a depth of about 1,500 meters about 8 hours ago. Maybe a bit less, maybe like 6 hours ago. And so far we've seen soft sediment and sand substrate, but very recently we started seeing these rocky outcrops that look like pillow basalt. And we're starting to see life, or big life rather, on it. There's plenty of life in the sediments, but big corals and sponges are appearing in the horizon. So we're gonna keep moving for a little bit towards our next waypoint, which is not very far away. To get a sense of what the community looks like here, and then I think it will be a good opportunity to start doing some collections of the diversity of this place.
Excellent. What are you most excited about seeing, sampling during this portion of the dive?
What this particular depth span is a depth in this region that hasn't been well explored at all. We did some work here in 2017, as well as in 2019. So 2017 with the Oceanus Explorer, 2019 with the Nautilus. We're mostly diving deeper or shallower than this depth range, so we're really gonna see some of the first observations of what the diversity is like in this region, in this dive. So it's very exciting, and obviously we're really close to shore, so it's very close to all the communities in American Samoa. So it'll be really interesting to see, you know, what's in their backyard right here of the island. So I'm very curious about also how the island may be providing some sustenance to all these creatures. So it should be a pretty interesting dive.
Mm-hmm.
Hey, pilot, can we take a look at
Yes.
this creature?
Yep.
Really?
Oh.
So Santiago, we're just constantly moving with the ship here, culling in continuous moves, but I'll check in with you if you'd like the ship to stop, and same with you if you're thinking that you want to sample or you want to hold here for more than a couple minutes, let us know and we'll hold position.
Roger, yeah, and I think I just want to take a quick look at this. I think I know this looks like a mushroom coral. Just want a confirmation. Most likely we'll continue to see them. So I don't think we need to stop the ship move.
Roger that.
Yeah, so this is a mushroom coral. It's an octocoral, possibly Antomastus or pseudo-Antomastus. This is a family of corals I'm really intrigued by because the close relatives of it make these really large colonies, sort of like trees. But this one never branches, so it stays like a little red ball with polyps. They call them mushroom because of that shape. Yeah. So we will zoom in. When they retract the polyps, they have a main stem and like kind of like it's called a capitulum. It's like the top of the mushroom. It's hard to imagine when they have the polyps out like that. Josh, could you log that observation as Anthomastus, possibly?
What was the scientific name again?
Anthomastus.
How do you spell that?
Athommastus.
Got it.
Thank you.
Beautiful specimen.
Yeah. So that is also interesting because we're pretty much within the depth range of the summit of Vailulu, which is an active volcano that we're very intrigued and we'll be hopefully visiting in a few days. So
get moving.
Be really also interested to see what the life at same level of the seamount looks like elsewhere. So we can compare it to what we see inside the crater of the volcano.
So as we move more upslope, will we see more potentially of these? Or are we kind of reaching the top of their range where we'd find these?
I don't think we know.
It's one of the questions.
So we'll see. Yeah, we've observed similar looking animals. It's really hard to tell what species they are. It could be the same genus but different species. But we've seen some very similar animals to this mushroom coral, a bit shallower as well. So I expect we'll continue to see them all the way to waypoint 3.
Cool.
Yeah.
And want to take a look at that? Push.
Hey pilot, if it's still possible to do a zoom, can we take a look at that one?
And get moving.
So still a bit hard to tell exactly which coral this is, but this looks like one of those corals I was describing as close relatives to the mushroom coral, to the one we were just seeing. So that obviously has branches, is like a little fan, but hopefully we'll get a look at a different specimen soon. And you can start seeing some sponges too. They look kind of like bases on these rocky outcrops. So this looks very much like volcanic rock. You see those pillow lavas that look kind of rounded. They are pretty typical um, eruptive rock.
And so would
That,
these species of corals be like pioneer species that are kind of the first in an area?
but this, yes and no. Some of the species that we're seeing here, some of the sponges are what we've seen in pretty old habitats. Yeah. So island was formed millions of years ago, right? And so those rocks are the product of those volcanic eruptions that happened a long time ago. And so, you know, we have that time span since the formation of the seafloor until now for these animals to have colonized and be growing there, but we don't know exactly when they arrived, right? And that's the case for most of the deep sea. But what's really interesting about an active volcano
I heard my
where
name but lost everything else.
these islands and other seamounts have been born, which is
DVL
where
reset? Yeah, I'm
Bailulu,
out
the
of
volcano
XY and
is that
depth. Yep.
we get to see what the fresh new habitat is, right? We know that's with only maybe a couple of years old. And so it's really interesting to compare really old habitat with really new habitat, because that gives us a sense of that, you know, whether which species come first, some maybe coming in at much later stages of the successional sequence in these benthic ecosystems.
Let's see, Jacob, while we're between spots, why don't you take a second to introduce yourself and kind of what your job is right now while we're moving through this dive?
I'm Jacob. I'm the data logger on the 4 to 8 shift for ROV watches. I'm pretty much recording everything interesting we're seeing, including different species, different geologic features, such as like basalt outcroppings, and also recording when we were taking samples such as eDNA samples using the multi-puffer or Niskins.
Hopefully we'll keep you really busy this dive with lots of organisms and species and samples to be cataloging.
Michael, is there anything that tells me what the indicator for the tilt on your camera is?
Yeah. So Michael, I think it'd be good for Navigator as well if you just let us know if you're going to be tilting, tilt or if you change heading, then we'll kind of like be able to keep everything in the box and monitor it a bit. Especially as Gabby gets, trained up. Yeah. Okay. Ship's still on the move. It has 10 more meters left.
Yeah, pilot. So I think the targets that we're after are the high ground returns on the sonar. Yeah.
Yeah. Looks like some's coming up in Atalanta shortly.
I see a question coming in from the audience, and I'm wondering the same thing. These semicircles that we see in the sediment, what, any idea what those are, what's making those?
They look biological, uh, given that they're mostly about the same size and shape. So maybe a creature that is making its home right there, or maybe it's feeding there. I don't know exactly who. There's people that are really good at telling which animals can make different marks in the sediment. I still have a lot to learn about that.
Maybe we'll see it later on.
Yeah, maybe somebody in the chat will have a suggestion.
How's it going, Tito?
And Herc.
That's correct. Yep.
Thanks.
And let me know if you need me to adjust gains there or anything. So yeah, what Tito's pointing out makes total sense. We've got Hercules has some little bubbly stuff over here to the right. We're seeing that there in the camera. And then in the general trend, Atalanta on the kind of a little bit upslope here. There'll be some more rocks.
Good question. So the ROV has laser scales.
Would you like to push on that feature?
I don't know exactly. We can ask the pilots in a minute. What
does the back table? What does the back table like
between them
lasers?
is? They're usually 10 centimeters, but we can ask them to turn them on.
So science row.
Yeah,
do you guys want lasers on?
I already turned them on.
They're on now.
Okay. Yeah,
They're
just
on now.
check, just checking.
Yeah.
See. Yeah.
Yeah. I was just explaining Ned back here.
Jana, would you push on that?
How do we tell the scale? So Ned, you see those green dots in the top of the screen? Yeah. So those are fixed distance and they're parallel to each other, and so they will give. We can use that as a scale. Naval pilot, do you know what's the distance between the two dots?
They are 10 centimeters.
Yep, 10 centimeters.
10 centimeters, yeah. Yeah, so that's pretty standard for ROVs.
Bridge nav.
10 centimeters.
One more step, 50 meters.
So that shrimp is about,
thank you,
8 centimeters, I would say. 8 to 10. So you see here, there's a lot of dead sponges. We'll see this a lot more. So this is pretty typical also of this region. I remember seeing this a lot in 2017, is that...
And moving right along.
There were a lot of sponge skeletons. Some of them were even encrusted in black mineral.
So we know by color, that's how we're identifying this?
Yeah, mostly. I mean, sponges can, like live sponges can have something that's colored like a yellowish and brownish. But these ones is both the color, but the appearance, they look like they are not, they might have some holes, or they might have some weird textures to it. So that's how I kind of, it's a telling sign for me of that. So we see another one of those pink fans in the bottom of the screen. And notice the orientation is perpendicular to the slope. So we're going uphill right now. But we want to keep an eye on that because that's the telling sign of what the currents are around here.
Interesting.
So I expect we'll see more colonies possibly in the same orientation, but we'll see. So you see there's more small sponges. You see some white, kind of like flowers coming out of the basalt. Those are sponges, very likely glass sponges. Oh look, there's another of those big pink fans coming up. Tito, is it possible at all to do a quick
I'm
scan of
gonna
this.
bring my heading over a bit, and I'll try to
there's
get that right in the
two,
center of the
actually.
screen.
There's two of them. There's one big one and one small one. You
It's
can
like
see
a smaller one in bottom right too.
Yeah, see three on the screen. So you can see from the scale that they are,
Jana, you want to push?
much bigger than the shrimp that we're just seeing. They're probably around 30 centimeters tall. And this looks like a bubblegum coral. But actually, it looks like it's growing on a bamboo skeleton, maybe? It's hard to tell. That I've never seen one doing that, so I think it's actually just a bubblegum coral fully. I got deceived by, I think, by the brightness of the stem. It seemed like a white skeleton. I thought I saw some bands, but no, this is clearly a bubblegum coral. So this is all very closely related to the mushroom coral. Pilot and co-pilot, how are we doing in moving for thinking about stopping for a collection?
I think that's a navigator question.
Yeah, sure. We are in position here. If I hold station, are you considering this species here, the specimen?
Yeah.
Okay, roger. I can hold station.
Thank
Bridge
you.
nav. Hold position.
So what's interesting is that entire
So
expedition of
monitor
2017
here.
of the Llanos, which we did several dives in the region, we didn't see a single specimen of this family of corals, or rather in this genus of corals. The bubblegum corals are some of the most common corals in the deep sea. But we have no idea what species this is. It might have been, maybe Steve Voskovich is in the chat and can tell us, maybe they saw them in the Nautilus in 2019. I don't recall, but those were
Yeah, spin around
longer
on
dives
the
and I
Cape,
haven't really sat
Cape
down and watched them
Hercules.
all. But now we have information of what it was seen in those from database of annotations that they had generated at the annotation laboratory at the University of Hawaii. So Megan Potts, who's the navigator here in the previous watch,
She did a great job of documenting all those for us.
Yeah, so she'll be able to tell us instantly.
MVP.
Fortunately, we can't keep her awake the whole time. But she gave me her spreadsheet, so I'm going to pull it up.
Santiago, are we planning on collecting that coral as a sample, or? Yeah, okay.
Yeah, that's the plan. They're positioning the vehicle so they can approach, so you can see they're guiding the vehicle to land on these hard rocks to do the collection. So it'll take a few minutes. Yeah. Yeah, that's a good idea. That's great. Yeah, good idea. Because we're starting to see a kind of more defined community of, we're seeing some of those mushroom corals, a few of these bubblegum corals, as well as some of those glass sponges bases, both living and dead. And so seems like a good place to take a eDNA sample. We collected eDNA samples earlier in the dive, throughout the dive. We were traversing over sandy bottoms of sediments, but now we're entering hard bottom habitat, and we're starting to see a lot of the animals that we recognize from this kinds of place. Okay, thank you, Steve. So Steve Voskovich in the chat is telling me that there were 2 samples of Paragorgia, which is the genus of this bubblegum coral, collected in 2019 with the Nautilus. So yeah, they did see them and collected them, which is great. But they were also around this depth. So we are, 1,300 meters, okay. It's possibly the same species.
So with that information, are you still wanting to collect a sample or?
I think we will still collect a sample. I would be curious to take a look at the one on the left, the smaller one too.
In the background on that rock?
Yeah, it just looks like the one that we saw earlier, but I couldn't really tell what it was. And it may be a little different from this bubblegum coral. It could be different species.
Sure.
And
Jada,
so,
would you push on that?
Also, hey Steve, this is Rennie. Just letting you know that Rubber Chicken has been working well and we have fulfilled its dreams in the moon pool.
Annette, did you hear Jacob? It's paused. Thank you. And what pump are we on? Pump 13 is paused. Thank you. So yeah, this one in the center screen now under the lasers looks different to me. The color, you see, it's a little bit more purple or pink. Are we full zoom, pilot?
That's full zoom.
That's full zoom, okay. Can we get any closer?
Oh, would
you see another species of coral in the back next to the lasers.
Yeah, the other one, I don't know, is that a Metallogorgia back there or was it some other kind of Chrysogorgia? I guess
yeah, that's what I was thinking. I guess we'll take a closer look. It looked like me, definitely like a coral in that family, Chrysogorgidae.
Yeah.
Or, I should check the new taxonomy. This is my colleague Andrea Quattrini and Kathy McFadden, my colleagues at the Smithsonian and Harvey Mudd College, published a really interesting paper recently that revamps the entire taxonomy of octocorals using the latest molecular information. So I think many of us in the community are really excited to have that out because it's a big, big, big contribution to our understanding of the diversity of life in the deep sea. And so we're trying to adhere to it because it's the latest new best standard. So I need to learn it completely. This is a great view. So you can see, well, you don't see it in the live stream, but I'm looking at one of the operation cameras, screens of the ROV that shows the heading. Yeah, so it has a compass, and we are pointing towards 90 degrees. So that's full east, and we can see the plane of the coral now. So that gives us confirmation that they're completely they're east-west facing, and this coral is indeed a different species. Is very likely a corallid in the genus Pleurocorallium or Corallium. Their sister or relatives, we always use the term sister in systematics to refer to species that are the most closely related to any other species. And in this case, these two groups of corals, the bubblegum corals and these are called precious corals, the one we're looking at, are sister groups of species. And then the mushroom coral is the next closest thing to these corals, right? So the three, it's amazing, the three first corals that we've seen are all in the same family, and they represent the diversity of large morphological changes in the evolution of life in that group. Thank you, Pilot, for that great look. I think this will be really good targets for collection, both of these, since we are sitting in what seems like a nice spot. What do you think? Do you see this as a good spot for sampling as we are positioned now, or do you need to reposition?
You're talking to the pilot?
Yeah, Tito.
Yeah, so we're only sampling with the port arm, I believe. Are you looking to get coral samples?
That's right.
Yeah, so the starboard arm with the coral cutters is not functioning.
But this is one of those lucky ones because these ones you don't need to cut them. They are, the one in the center is brittle, so it will break really easily.
Alrighty, I think
And
I need to come around from the other side. But,
yeah,
yeah, give me a
and
minute.
then the one on the left is that's a spongy tissue, and so with any or little pressure, just twisting it, it will break off. It's not as brittle, but it is brittle enough that you don't need the coral cutters.
Yeah, it may be difficult to sample a coral with that arm. It's
Yeah.
not really. It kind of smashes it, and the way that the arm move, I can't imagine that going well.
So it's a rate arm that we're using the GUI to operate, and I think, have you run it yet, Michael?
I think.
So the craft arm is down. I wasn't aware of that. That's fully down. Okay. Yeah, I don't. I wouldn't imagine this will go well.
No, I'm not sure
because basically each command it just is going to jerk towards it, and then it'll smash it. I don't know that we would get a viable sample.
Well, but that's going to be true of any sample we want to do in this dive, right?
That's true, except for rocks. Yeah.
So I would still be really interested in giving it a try because we want to see if we can collect samples at all in this dive.
Okay. So we'll position for it.
Well, I think
and then
what would be wise would be maybe starting with a little practice with grabbing a rock.
Roger.
So there's value in learning that fact.
Yeah, you'll get a sense for what we're working with, and if you think that's viable to check out with the coral.
Sounds good.
And we don't have to actually collect the rock. If you don't want to, we can set it down. But
just a little practice with the arm and a demonstration. So
sounds good.
We haven't yet, but I was thinking that once we settle, before we collect the coral, we could try to fire the Niskin. So we have kind of the picture of the community, not the picture of the event that will disturb the community a little bit.
I don't.
So Michael, how about reaching out and trying to grab one of these?
Pilot. Another idea also for when we are activating the manipulator is that this is a place where it would be good to take a Niskin bottle. Yeah, I agree. They did manage to collect one in the, yeah. Do you know if they managed to get that one just because it was the only one that they could get?
Yeah, we could give it a shot.
Yeah, I guess
as long as everyone's patient.
Yeah, that's fine. Yeah, I'm not sure what was the reason that one was chosen. If it was just because it was the only one to reach, or it was kind of a routine one. Yeah, but I completely understand, co-pilot, your point. Yeah, if we can, we can. If we can't, we can't. We just want to give it our best to get the most out of the dive.
Would you like me to do things like the jaw while you're doing the moving? Let's see, you know, we're just experimenting here.
In Santiago, we are near waypoint 3 of the dive.
We are, yeah, about 80 meters from it. So yeah, I'll call this associated with waypoint 3.
Got it.
Just come down some more, shoulder down.
In the vicinity of waypoint 3.
What's that? Shoulder up.
You got a rock. We might as well collect it if you have it in the claw, if you are in a good spot for that.
So I'm hearing some noise about they may want to put that in the box. You're making it look so easy so far.
Yeah, I'm getting more and more excited about collecting.
I believe they may have collected one rock sample so far.
Have they collected anything?
I'm not sure. It had to have gone forward biobox.
In the boxes?
So let's also take into consideration the only biobox we have available is the forward biobox.
So back row, do you guys hear that? As we're talking about this, because we don't have that starboard arm, we do only have that forward bio box.
Which has 2 compartments in it.
2 compartments.
Roger.
Yeah, so
that's fine.
We can obviously collect, but just to keep in mind that yeah, or we can attempt to. That's what Michael's going to try to do.
Yeah. Attempt to is what we can do. Yeah, thank you for the information. I see a milk crate in the front, right? That was for rocks, is that right?
That is for the weights, ballast weights. And if it's not quite empty, but we'd have to pull a weight out later, I mean, as we collect the rocks.
Is there only one way in there?
I can't count right now, but it looks like there's at least one.
Okay. Do you think there's a chance that they can share with the potential risk that we might lose it?
I think if the weight is if the weights are eventually emptied, perhaps, but otherwise you could potentially, if you were to put one in there and then later go to pull the weight out, that you could either lose the sample or it could get mixed up.
Yeah, I'm just thinking that we don't have anywhere else to put it unless we put it in the bio box, which I guess is also good.
Well, that's up to you guys.
Worst thing that can happen is we break it, you know, or
yeah.
damage it. So
sure.
No point in not doing it. So basket all the way out.
Yeah,
this sample of rock, you can see has a dark coloration, very dark.
I'm seeing like 2 weights in there.
Wow, that's fast.
Iron manganese oxide.
So looks like 2 or 3 plates in there.
Primarily manganese.
Well, you know, just to show off, why don't we put the rock down on the porch and pick out a weight?
It's actually very likely
get rid
that
of it, put
the
rock
actual
in there.
underlying volcanic
I'm
rock
on
is
the
very
border of
small,
heavy, so I would
and
mind
it's
doing that.
heavily encrusted with that mineral.
If there's anything you want me
But
to
we'll
do
find
as
out.
far as
Okay.
jaw
Crack it
or
open.
port. So I'm going to say start
Looking
off
forward
with
to
shoulder
taking
rotate.
rock slices when we get these
See what
samples.
this
Yeah, that's right. We have a saw on board.
Come in
We
just
have
a
a
little
rock
bit.
saw on board, so we
Rotate
can
the other way.
get a nice cut with it. Jacob, are you gonna be doing that?
Maybe. We'll see.
So
that
back row, I
sounds
don't know
fun.
if you copied that. We're going to place the rock on the porch, remove a weight, and then we're going to put the rock in the basket.
Good plan.
Yeah.
You're
Jacob, which
getting
sample number
Yeah,
is
it's
this?
showing off.
I like it.
Sorry, say that again.
Which sample number do we have here?
This would be 8.
Sample 8. Okay. I will have to
Sample 8.
talk with my team. We have not been,
What have been
dropping enough
samples
sample
1 through
targets.
7?
Yep. Samples 1 through 7 are done. Sample 7 was the multi-puffer, so.
Oh, okay.
Right.
You have
Copy
multi-puffer,
that,
okay.
of course.
I'm going to go ahead and release the rock right there.
So mostly all the samples except for, I don't know, I guess all the samples have been EDNS so far, right?
And let's go
This is the
after a weight.
first. We've had
non-EDNS.
I think we just had one Niskin that was